Could a single daily habit support your longevity while simultaneously presenting a significant challenge to your sleep quality? Recent viral discussions on social media and emerging longevity research have brought this exact question into sharp focus. While moderate coffee consumption is associated with promising cellular mechanisms supporting longevity, the active caffeine in your bloodstream can have a notable impact on sleep architecture. I am your science communicator at Beyond Limit Health, exploring these pathways while recognizing individual variability and the current limits of evidence. Today, we are bypassing subjective trends to look directly at the molecular pathways, pharmacogenomics, and chronobiology of coffee. The chronobiology of longevity, the 2025 morning type discovery. To understand coffee's impact on lifespan, timing is crucial. A breakthrough study published in January 2025 in the European Heart Journal analyzed 40,725 adults from the NHA-NES cohort and validated the findings in the Women's and Men's Lifestyle Validation Study. The researchers identified that a morning-type coffee drinking pattern, consuming coffee predominantly in the morning, was significantly associated with lower risks of all-cause mortality, hazard ratio 0.84, and cardiovascular disease-specific mortality, hazard ratio 0.69, compared to non-coffee drinkers. Conversely, the all-day type pattern showed no significant association with reduced mortality risk. This suggests that afternoon or evening consumption might disrupt critical circadian rhythms, such as melatonin secretion, and elevate cardiovascular risks. Beyond chronological time, how does morning coffee slow biological aging? A recent analysis of NHA-NES data suggested that moderate coffee intake was associated with modestly slower biological aging acceleration as measured by epigenetic clocks. At the molecular level, epigenome-wide association studies reveal that coffee consumption alters DNA methylation in blood cells. Most notably, a specific site, CG21566642, near the ALPPL2 gene, exhibits a highly significant demethylation signature, which helps suppress genes associated with systemic chronic inflammation. Additionally, coffee activates a vital cellular cleanup process called autophagy. Landmark research from the Guido Kromer lab demonstrated that both regular and decaffeinated coffee trigger rapid autophagic flux in the liver, heart, and skeletal muscle of animal models within 1 to 4 hours of ingestion. This response is driven by the inhibition of mammalian target of rapamycin complex 1 and a global deacetylation of cellular proteins, prompting cells to clear out toxic protein aggregates and dysfunctional mitochondria. However, this biological benefit comes with a sleep trade-off. During wakefulness, the neurotransmitter adenosine builds up in your brain, binding to A1 and A2A receptors to generate homeostatic sleep pressure. Caffeine, acting as a competitive antagonist, crosses the blood-brain barrier and binds directly to these receptors. It does not eliminate fatigue, it merely blinds your brain to the accumulated sleep drive. Quantitative EEG analyses show that even if you fall asleep, caffeine compromises sleep microstructure. It suppresses slow-wave activity, specifically delta oscillations, in the 0.5 Hz to 4.5 Hz range during non-REM stage 3, N3 sleep, which is the critical physical recovery window for waste clearance in the brain. A meta-analysis shows that daytime caffeine reduces total sleep time by an average of 45 minutes, decreases N3 deep sleep by 11.4 minutes, and delays REM sleep. This can initiate a potential vicious cycle where morning fatigue leads to heavier daytime reliance on caffeine. Why does sensitivity vary so widely? Approximately 95% of caffeine clearance is regulated by the liver enzyme CYP1A2 and the SNPRS 762551 within its genes splits us into distinct phenotypes. In slow metabolizers, AC or CC carriers, representing approximately 59% of the population, higher coffee intake is associated with significantly increased odds of myocardial infarction due to prolonged vasoconstriction. Conversely, fast metabolizers, AA genotype, approximately 41%, show no increased MI risk and, in some analyses, a protective trend. Sensitivity is further modulated by the master switch gene AHR, RS441070, and your brain's receptor sensitivity gene ADORA2A, RS5751876. There is also a fascinating glucose paradox with coffee. Acutely, a single dose of caffeine impairs postprandial insulin sensitivity by antagonizing muscle A1 receptors, leading to temporary blood glucose spikes. Yet, long-term epidemiological studies consistently show that habitual moderate coffee consumption is associated with a 20% to 30% lower risk of developing type 2 diabetes. This chronic benefit is driven by non-caffeine polyphenols like chlorogenic acids, which delay glucose absorption in the gut and protect insulin-secreting beta cells. Furthermore, Mendelian randomization analyses show that filtered coffee enriches the gut probiotic genus Veillonella, which produces propionate, a short-chain fatty acid that enhances insulin sensitivity and directly lowers glycated hemoglobin. To capture the promising cellular benefits of coffee without sabotaging your sleep, implement this precise, evidence-based chronobiological protocol. First, delay caffeine intake after waking. One emerging chronobiological strategy, recommended by some researchers, is to delay the first cup 60 to 120 minutes after waking, allowing the natural cortisol awakening response peak to subside before introducing caffeine. This prevents overstimulating your HPA axis, though individual response varies significantly. Second, respect the sleep boundary. To avoid reductions in total sleep time and slow-wave suppression, systematic pharmacokinetic analysis suggests stopping standard drip coffee, containing approximately 107 milligrams of caffeine, at least 8.8 hours before bed. If consuming high-dose formulas, approximately 217.5 milligrams, clear it at least 13.2 hours before bed. Third, if you are a genetic slow metabolizer, choose decaf. Preclinical studies from the Cromer lab demonstrate that decaffeinated coffee triggers autophagy and epigenetic benefits identically to regular coffee, with none of the caffeine-related sleep or cardiovascular liabilities. Coffee is not a mindless stimulant, but a powerful biohacking tool that requires precise scientific calibration. Real health optimization is about understanding molecular mechanisms and respecting your biology while recognizing individual variability and the current limits of evidence. The content provided today is for informational purposes only and is not medical advice. Please consult with a healthcare professional before altering your habits. If you are ready to rationally optimize your body and mind within a scientific framework, subscribe to Beyond Limit Health and share this with a fellow coffee drinker. We will see you in the next video.